Hey everyone, welcome to For Science Salah Lotfy channel. Today, we're diving into a fascinating topic that's crucial for cotton farmers everywhere: how the pink bollworm, or Pectinophora gossypiella, has become more resistant to insecticides. This tiny pest has some serious tricks up its sleeve, and understanding them can help us develop better ways to protect cotton crops. First, a bit of history. The pink bollworm has been wreaking havoc on cotton fields since the early 20th century. It burrows into cotton bolls and causes major damage, leading to huge losses in yield. To fight back, farmers have traditionally used insecticides. But over time, these pests have adapted, making chemical treatments less effective. In our latest research, we took a closer look at this resistance. By studying 4 different field populations of pink bollworms and comparing them to a lab strain, we found some interesting results. The field populations had higher levels of 2 detoxifying enzymes, glutathione-S-transferase or GST, and beta-esterases. These enzymes help the pink bollworms break down insecticides, allowing them to survive treatments that would otherwise be lethal. Next, we used real-time PCR technology to investigate the genetic side of things. This powerful tool helps us measure how much certain genes are expressed. We focused on the BtR gene, linked to Bt toxin resistance, and the Cad1 gene, involved in general insecticide resistance. Both genes were more active in the field populations compared to the lab strain, confirming that these pests are biologically equipped to handle chemical threats. So, what does this mean for cotton farmers? Understanding these resistance mechanisms is key to developing better pest management strategies. By implementing integrated pest management, or IPM programs, farmers can reduce their reliance on insecticides and use them more effectively. This approach is especially important for countries like Egypt, where cotton is a critical part of the economy. On a global scale, resistance to insecticides is a major challenge for agriculture. The methods and findings from this study can be applied to other crop pests, helping to enhance food security and minimize economic losses worldwide. Alright, that's it for today. Remember, continuous research is vital for staying ahead of these crafty pests. Stay tuned for more scientific insights right here on For Science Salah Lotfy channel.
Welcome to For Science Salah Lotfy channel. Today I'm diving into an eye-opening study on insecticide resistance in a notorious cotton pest, the pink bollworm. This little critter has been outsmarting us for years, but new research is shedding light on how detoxifying enzymes and gene expression are helping them win the battle. Let's dive into it. The study, titled Detoxifying Enzymatic Activity and Insecticide Resistance Gene Expression in Field Populations of Pink Bollworm, was published in the Arab University's Journal of Agricultural Sciences in 2021. It was received in September and accepted by December. Quite the rapid turnaround for such groundbreaking work. This research comes from a stellar team: Dr. Rabab, Abdelaziz, Desouki, Allam from the Bollworms Research Department, Plant Protection Research Institute, and Professors Dr. Amged Mohamed Kamal Sobeha and Professor Dr. Abdelaziz Abouelela Kidder from Ain Shams University. And let's not forget Professor Dr. Naima Kotba Al-Senoui, also from Ain Shams University. Quite the dream team, right? Now let's talk pink bollworms. These pests have been a menace to cotton crops for over a century. They infest cotton bolls, causing devastating yield losses. Farmers have been fighting back with insecticides, but resistance is becoming a bigger problem every year. This study digs into the biochemical and genetic tricks these pests are using. Researchers compared 4 field populations of pink bollworm to a lab strain. The field populations showed higher levels of glutathione-S-transferase, GST, and beta-esterases. These detoxifying enzymes are like the pest's secret weapons against insecticides. Using real-time PCR technology, the study also looked at resistance-related genes. The BtR gene, linked to Bt toxin resistance, showed different levels of expression across populations. Similarly, the Cad1 gene, associated with insecticide resistance, had increased expression in field population. That's some impressive survival strategy. These findings are a game changer. By understanding how resistance works, we can develop more effective pest control strategies. Integrating biochemical analysis with genetic studies gives us powerful tools for integrated pest management, or IPM. This helps farmers make smarter pesticide choices and slows down resistance development. This research is vital not just for Egypt, where cotton is a major crop, but also for global agriculture. The methods used can be applied to other crop pests, supporting sustainable farming and reducing economic losses worldwide. That's a win for everyone. Scientific research is key to securing our agricultural future. Thanks for tuning into For Science Salah Lotfy channel. Stay tuned for more amazing discoveries.
Cotton, a fabric so ubiquitous, so familiar it's easy to forget its origins. This humble plant with its fluffy white bolls has clothed humanity for millennia. From the clothes we wear to the sheets we sleep on, cotton is an integral part of our lives. Its fibers, spun into thread and woven into fabric, provide comfort, durability, and versatility. The global demand for cotton is immense. Millions of farmers depend on this crop for their livelihoods. The journey from field to fabric is a complex one, involving numerous stages of processing and production. However, this vital industry faces a constant threat: the pink bollworm. This tiny insect, barely visible to the naked eye, poses a significant risk to cotton production worldwide. Understanding this pest and finding effective ways to manage it is crucial for the future of cotton farming. The pink bollworm is a formidable foe. Its larvae burrow into cotton bolls, devouring the developing seeds and damaging the fibers. This infestation reduces yield and diminishes the quality of the cotton, leading to significant economic losses for farmers. The pink bollworm's notoriety as a cotton pest dates back over a century. Its arrival in the United States in the early 1900s marked a turning point in cotton farming. Initially, insecticides seemed like the solution. Farmers sprayed their fields, hoping to eradicate the pest. However, the pink bollworm proved to be a resilient adversary. Over time, it developed resistance to many commonly used insecticides. This resistance, a result of natural selection, meant that the surviving insects passed on their ability to withstand the chemicals to their offspring. Consequently, farmers faced an uphill battle against an increasingly resistant pest. To combat the pink bollworm effectively, scientists needed to understand the mechanisms behind its insecticide resistance. A recent study delved into the insect's biology, focusing on detoxifying enzymes and insecticide resistance genes. These enzymes present in the insect system can break down or neutralize insecticides, rendering them ineffective. The study aimed to analyze the levels of these enzymes in pink bollworm populations and correlate them with the presence of specific genetic markers associated with resistance. By understanding how these enzymes and genes work in tandem, scientists could potentially develop new strategies to overcome insecticide resistance. The study involved collecting pink bollworm samples from different geographical regions, representing a range of insecticide exposure histories. This approach allowed researchers to compare enzyme activity and genetic profiles across populations with varying levels of resistance. Section 4. Adaptability. The pink bollworm's secret weapon. The study's findings provided valuable insights into the pink bollworm's remarkable adaptability. Researchers discovered a strong correlation between high levels of specific detoxifying enzymes and the presence of known insecticide resistance genes. This finding confirmed that these enzymes play a crucial role in the insect's ability to survive insecticide exposure. Furthermore, the study revealed variations in enzyme levels and genetic profiles among different populations. This geographical variation highlights the dynamic nature of insecticide resistance. Pink bollworm populations, constantly evolving in response to their environment, adapt to local insecticide use patterns. These findings underscore the limitations of relying solely on insecticides for pest control. The pink bollworm's ability to develop resistance so effectively necessitates a more sustainable and multifaceted approach. Section 5. Fighting back. A balanced approach to pest control. The study's results have significant implications for pest management strategies, particularly integrated pest management, or IPM. IPM emphasizes a holistic approach, combining various pest control methods to minimize environmental impact and reduce the risk of resistance development. Understanding the specific mechanisms of insecticide resistance in the pink bollworm allows for the development of more targeted and effective control measures. For example, knowing which detoxifying enzymes are prevalent in a particular population can guide the selection of insecticides that are less likely to be broken down. Furthermore, This knowledge can inform the development of new insecticides that target different pathways in the insect, circumventing existing resistance mechanisms. By integrating these findings into IPM strategies, farmers can move towards more sustainable cotton production, minimizing their reliance on insecticides and preserving the longevity of this vital crop.
The pink bollworm is a dangerous pest. It attacks cotton crops. Farmers use insecticides to protect their crops, but the pink bollworm is becoming resistant to these insecticides. This is a growing problem. It makes it harder to control the pest. Scientists are working to understand how resistance develops. This knowledge will help us find new ways to manage the pink bollworm. Resistance is a serious threat to cotton production. We need to find sustainable solutions. Insects have natural defenses against toxins. Enzymes are one such defense. They can break down insecticides. This makes the insecticide harmless to the insect. Two important enzymes are beta-esterases and GST. Beta-esterases break down certain types of insecticides. GSTs help remove a wide range of toxins from the insect's body. When insects develop resistance, they produce more of these enzymes. This allows them to survive exposure to higher doses of insecticides. Genes control an organism's traits. This includes its ability to resist insecticides. Scientists study gene expression to understand resistance. Gene expression is the process of using genetic information to make proteins. Proteins carry out various functions in the cell, including the production of enzymes. By studying which genes are more active in resistant insects, scientists can identify the mechanisms of resistance. Understanding these mechanisms is crucial for developing new strategies to overcome insecticide resistance. Section 4. Inside the laboratory, studying resistance at the molecular level. Scientists use a technique called real-time PCR to study resistance. This powerful method allows them to quantify the presence of specific RNA molecules in a sample, providing crucial insights into gene expression. This technique measures the amount of specific RNA molecules in a sample. By doing so, it helps researchers understand the molecular mechanisms underlying resistance. RNA carries genetic information from DNA to the protein synthesis machinery. This transfer of information is essential for the production of proteins that can confer resistance. The process involves several steps. Each step is meticulously planned and executed to ensure accurate results. First, RNA is extracted from the insects. This step is critical as it provides the raw material needed for subsequent analysis. Then, specific primers are designed to target the genes of interest. These primers are short sequences of nucleotides that bind to the target RNA. Finally, PCR amplification is performed, and the results are analyzed to determine the level of gene expression. This step amplifies the target RNA, making it easier to detect and quantify. This method helps scientists understand how gene expression changes in response to insecticide exposure. By analyzing these changes, researchers can develop strategies to combat resistance and improve pest control methods. Section 5. From Field to Lab. Understanding resistance in pink bollworm. Research shows that pink bollworm populations in the field have developed varying levels of resistance. This resistance is a significant challenge for cotton farmers and researchers alike. Some populations are highly resistant, making it difficult to control their spread and impact on crops. Others show moderate or low resistance, which still poses a threat but can be managed with current methods. Laboratory strains of pink bollworm also show resistance, indicating that this trait is not limited to field populations. However, resistance levels are often higher in field populations, suggesting that environmental factors play a crucial role. This suggests that field populations are under stronger selection pressure, which accelerates the development of resistance. Factors like continuous insecticide exposure and limited gene flow contribute to higher resistance levels in the field. These conditions create an environment where only the most resistant individuals survive and reproduce. Understanding these differences is key to developing effective resistance management strategies. By studying both field and laboratory populations, scientists can devise better methods to combat resistance and protect cotton crops. Section 6. A call to action: sustainable cotton farming for the future. Insecticide resistance is a significant challenge that cotton farmers face today. This resistance is primarily due to the overuse of chemical insecticides, which has led to the evolution of stronger, more resilient pests. It threatens the sustainability of cotton farming, making it harder for farmers to protect their crops and ensure a good yield. We need to find new ways to manage the pink bollworm, one of the most destructive pests in cotton farming. Integrated pest management, or IPM, is a holistic approach that combines various strategies to control pest populations effectively. It combines different methods to control pests, reducing the reliance on chemical insecticides and promoting sustainable farming practices. These methods include crop rotation, biological control, and the use of pest-resistant cotton varieties. By using resistant cotton varieties, farmers can significantly reduce the impact of pests on their crops. By adopting IPM strategies, farmers can reduce their reliance on insecticides, which in turn helps to preserve the environment. This will help to slow down the development of resistance, ensuring that insecticides remain effective for longer periods. It will also promote a healthier environment, benefiting not just the farmers, but the entire ecosystem. Sustainable cotton farming is the way forward for a greener, more resilient future.
The silent threat to cotton. A story of the pink bollworm. Cotton, a plant we often take for granted, is a vital part of our lives. From the clothes we wear every day to the soft sheets we sleep on at night, cotton is woven into the fabric of our daily existence. It provides the comfortable clothes we wear and the soft sheets we sleep on. Cotton is not just a crop, it's a cornerstone of comfort and utility in our lives. Yet this precious resource is under constant threat from a tiny but formidable foe, the pink bollworm. This pest, though small in size, has a massive impact on cotton production. Imagine a creature so small it can slip unnoticed into a cotton boll, wreaking havoc from within. The pink bollworm's ability to infiltrate and destroy cotton bolls makes it a silent but deadly adversary. This is the story of the pink bollworm, a pest that has plagued cotton farmers for centuries. Its history is intertwined with the history of cotton farming itself, marking a long-standing battle between man and pest. The pink bollworm, scientifically known as Pectinophora gossypiella, is a moth larva that feeds on the cotton plant's seeds and fibers. Its life cycle is perfectly adapted to exploit the cotton plant, making it a persistent threat. Its presence can devastate cotton yields, impacting livelihoods and economies across the globe. The damage caused by this pest can lead to significant financial losses for farmers and affect the global cotton supply chain. Understanding this tiny creature and its remarkable ability to develop resistance to insecticides is crucial for protecting our cotton crops. Researchers and scientists are constantly working to find new ways to combat this resilient pest and safeguard the future of cotton farming. Enzyme activity, a tale of two bollworms. In the heart of Egypt, a fascinating study unfolded, revealing the secrets of the pink bollworm's survival tactics. These tiny pests have long been a bane to cotton farmers, causing significant damage to crops and economic losses. Scientists ventured into cotton fields, collecting pink bollworm samples from different regions. Their mission was to uncover the hidden mechanisms that allow these insects to thrive despite the widespread use of insecticides. These field-collected insects were then compared to their counterparts raised in a controlled laboratory environment. This comparison was crucial to identify any differences in their biological responses. The goal to understand how exposure to insecticides in the field affects the bollworm's enzyme activity. By analyzing these differences, researchers hope to develop better pest management strategies. Enzymes, the workhorses of the biological world, play a crucial role in an organism's response to its environment. They are involved in countless biochemical processes that sustain life. In the case of the pink bollworm, two enzymes, GST and beta-esterases, took center stage in this scientific drama. These enzymes were found to be particularly important in the bollworm's ability to detoxify harmful substances. These enzymes are known to be involved in detoxification processes, helping insects break down harmful substances like insecticides. Understanding how these enzymes work could lead to new ways to combat these resilient pests, ensuring the protection of cotton crops for future generations. GST activity, a geographic perspective. The study's findings painted a fascinating picture of the pink bollworm's adaptability. GST activity, a key indicator of insecticide resistance, varied significantly across different Egyptian governorates. This geographic variation suggested that bollworms in areas with a history of intensive insecticide use had evolved higher levels of GST activity. This adaptation allowed them to survive and reproduce, passing on their resistance traits to their offspring. For instance, bollworms collected from the Dakalia governorate, known for its high insecticide use, exhibited significantly higher GST activity compared to those from the Fayum governorate, where insecticide application was less frequent. This geographical variation in enzyme activity highlighted the powerful selective pressure exerted by insecticides on insect populations. beta-esterase, the resistance enzyme. The story of beta-esterase added another layer of complexity to the pink bollworm's resistance mechanisms. This enzyme, known for its role in breaking down a wide range of insecticides, was found to be elevated in field-collected bollworms compared to their laboratory-reared counterparts. This finding suggested that exposure to insecticides in the field had triggered an increase in beta-esterase activity, bolstering the bollworm's defenses. This increase in beta-esterase activity is a classic example of how insects evolved to counter the threats posed by insecticides. By producing more of this enzyme, pink bollworms could effectively neutralize the toxic effects of certain insecticides, ensuring their survival. Section 5. Unmasking resistance. The genes tell the story. Delving deeper into the pink bollworm's genetic makeup, the researchers uncovered further evidence of their adaptive prowess. By meticulously analyzing the genetic sequences, they were able to pinpoint specific changes that had occurred over time, allowing these pests to survive in increasingly hostile environments. They focused on two genes, BtR and Cad1, known to be associated with insecticide resistance. These genes were not just randomly selected. Previous studies had hinted at their crucial role in the pink bollworm's survival strategy. BtR is involved in resistance to Bt toxins, a type of biopesticide, which is derived from a naturally occurring bacterium. This biopesticide has been widely used in agriculture to control various pests, while Cad1 plays a role in resistance to pyrethroids, a class of synthetic insecticides. Pyrethroids are commonly used due to their effectiveness and relatively low toxicity to humans and animals. The results were striking. Field-collected pink bollworms, those exposed to a cocktail of insecticides in their natural environment, showed a remarkable ability to adapt. They showed significantly higher expression levels of both BtR and Cad1 compared to their laboratory-raised counterparts. This difference in gene expression was a clear indicator of the evolutionary pressure exerted by the insecticides. This upregulation of resistance genes provided a genetic explanation for the observed increase in enzyme activity. The enzymes produced as a result of these genes helped the bollworms detoxify the insecticides more efficiently, and the bollworm's ability to withstand insecticide exposure. This genetic insight is crucial for developing new strategies to manage resistance and protect crops from these resilient pests. Section 6. Implications for pest management, a changing landscape. The findings of this study send a clear message. The pink bollworm is a formidable adversary, constantly evolving to overcome the challenges we throw its way. Our reliance on insecticides, while seemingly effective in the short term, is driving the evolution of resistance in insect populations. This arms race between pest and pesticide has significant implications for pest management strategies. Traditional approaches, heavily reliant on chemical control, are becoming less effective. We are witnessing a decline in the efficacy of insecticides, forcing us to rethink our pest management strategies. The pink bollworm story serves as a stark reminder that we need to adopt more sustainable and integrated approaches to pest management. Section 7. Sustainable pest management, a call for action. The challenge posed by insecticide resistance demands a paradigm shift in our approach to pest management. We must move away from our dependence on chemical solutions and embrace more sustainable and environmentally friendly practices. This shift requires a multifaceted approach, combining a range of strategies to effectively manage pest populations while minimizing our ecological footprint. Integrated pest management, or IPM, offers a promising solution. This holistic approach emphasizes the use of a combination of methods including cultural practices, biological control, and the judicious use of pesticides as a last resort. By adopting IPM strategies, we can disrupt the pink bollworm's life cycle, reduce reliance on insecticides, and promote a healthier and more balanced ecosystem. Section 8. The future of cotton, a shared responsibility. The future of cotton, a crop vital to our lives, hinges on our ability to adapt and innovate. We must move beyond the short-sighted approach of relying solely on insecticides and embrace a more sustainable and holistic vision for pest management. This requires a collective effort, involving scientists, farmers, policymakers, and consumers alike. Researchers continue to explore new avenues for pest control, such as developing novel biopesticides, breeding resistant cotton varieties, and harnessing the power of gene editing technologies. Farmers are at the forefront of implementing these innovations, adopting IPM strategies and embracing more sustainable farming practices. Section 9. Conclusion: Adapting to survive. The story of the pink bollworm is a powerful reminder of nature's resilience and the unintended consequences of our actions. It highlights the urgent need for a fundamental shift in our approach to pest management. By embracing sustainable practices, investing in research and innovation, and working together, we can ensure the health of our crops, protect our environment, and secure a sustainable future for cotton and the countless people who depend on it. The pink bollworm may be a formidable foe, but we possess the ingenuity and determination to outmaneuver it and ensure a thriving future for this essential crop. Welcome to For Science Salah Lotfy channel. Today, we're diving into some fascinating research on how the pink bollworm, Pectinophora gossypiella, outsmarts insecticides. This tiny pest has become a formidable adversary for cotton farmers worldwide, causing significant economic losses and challenging pest control measures. This pesky little critter is a major headache for cotton farmers. Its ability to develop resistance to various insecticides makes it a particularly tough nut to crack. So let's break down the science behind its resistance. Understanding the mechanisms at play can help us develop better strategies to manage this pest. Researchers took a close look at two key enzyme systems in field populations of pink bollworm from different governorates in Egypt. These enzymes play a crucial role in the insect's ability to detoxify insecticides. First up, glutathione-S-transferases or GSTs. These enzymes are part of the detoxification system that helps the pink bollworm neutralize harmful substances, including insecticides. These enzymes detoxify harmful substances, making it easier for the pink bollworm to survive insecticide exposure. The study found that GST activity was significantly higher in field strains compared to a laboratory strain. This suggests that the field populations have adapted to the presence of insecticides in their environment. Interestingly, the Fayum population showed the highest activity, while Columbia had the lowest. This variation indicates that local environmental factors might influence enzyme activity levels. Next, we have non-specific beta-esterases. These enzymes are another line of defense for the pink bollworm, helping it resist organophosphate and pyrethroid insecticides. These enzymes help the pink bollworm resist organophosphate and pyrethroid insecticides, which are commonly used in pest control. The research showed that all field strains had higher esterase activity compared to the lab strain, with Fayum again topping the chart. But wait, there's more. This higher activity level indicates a robust mechanism for detoxifying insecticides. The researchers didn't stop at enzymes. They wanted to understand the genetic basis of this resistance. They used real-time PCR, RT-PCR, to measure the expression of two key resistance-related genes. This technique allows for precise quantification of gene expression levels. The BtR gene, which is linked to resistance against Bacillus thuringiensis, Bt toxins, and the Cad1 gene, a receptor protein also involved in Bt resistance. These genes are crucial for the pink bollworm's survival in Bt cotton fields. The results? Both genes were upregulated in field populations, suggesting a genetic adaptation to insecticide exposure. This upregulation indicates that the pink bollworm is evolving at a genetic level to withstand insecticides. This research underscores the importance of sustainable pest management strategies to delay resistance development. Integrated pest management (IPM) approaches can help mitigate the impact of resistance. A big shout out to the incredible researchers behind this study. Their dedication and hard work are paving the way for more effective pest control solutions. Dr. Rabab, Abdelaziz, Desouki, Allam, from the Almondworm Research Department, whose expertise in entomology has been invaluable. Dr. Amged Mohammed Kamal Sobeha, Professor Emeritus of Pesticides Chemistry and Toxicology, whose insights into pesticide chemistry have greatly contributed to this research. Dr. Naima Kotba Al-Senaway, Associate Professor of Genetics, whose genetic analysis has shed light on the molecular mechanisms of resistance. And Professor Dr. Abdelaziz Abouelela Khider, Head Research Emeritus of Pesticides Chemistry, whose leadership has guided this groundbreaking study. Thanks for watching for Science Salah Lotfy channel. We hope you found this deep dive into insecticide resistance as fascinating as we did. Stay tuned for more insightful research discussions. Until next time, keep exploring the wonders of science.